there's a lot of time this notion that really successful scientists have to have this like eureka moment in their childhood. You know, you hear these stories of Nobel laureates who got their first chemistry set at 10 years old, and then they knew from there that that was kind of their destiny. But I really think that this isn't the case for a lot of people, and it wasn't the case for me, right? As a kid, I was smart and I liked science, but I also really liked art and literature and music and video games and reading and everything on the gambit. And so a lot of times throughout my career, I kind of felt that I almost fell into science rather than it was, you know, this predetermined thing that I knew since I was a young child.
This path you had to take.
Right, yeah. And I think that that is absolutely okay and should be almost talked about more, because I think a lot of young scientists feel this pressure that they're like, oh my gosh, I didn't know I wanted to be a scientist until halfway through my undergraduate career.
Right.
But that was how I ended up. So I think the most interesting story about how this entire career can be also kind of spontaneous was how I got into research. So I started my undergraduate career as a biologist because I really liked animals as a kid, and I'm like, maybe I should become a veterinarian, you know, who wouldn't want to play with animals every day? And as a biologist, you have to take your intro organic chemistry class, which most people despise.
I remember that one, and I despised it myself.
But I loved it, and I did really well in it. And so in one of the summers between semesters, I just ran into my organic chemistry professor at a cafe, and she remembered me because I did well in the class, and she offered me a lab in her, or a position in her research lab. And I didn't really know what research was, and so I was like, yeah, sure, I'll try it out. But it was, you know, I fell in love instantly. It was something that I felt that I was really good at. It really stimulated me. It was extremely rewarding, but I didn't change my major to chemistry, and I didn't know that I wanted to do research until halfway through my undergraduate career. And so, yeah, like I said, I sort of just kind of fell into the science and fell in love with it, and it wasn't a love at first sight sort of deal. And I really dislike this notion that people kind of have to be these scientific savants as kids and making discoveries before they can, you know, like drive or drink. Because to be honest, when I was a kid, I think I was more invested in beating the latest Zelda game than actually concentrating on my schoolwork.
Yeah, but that's how you learned how to like good problem solving.
Exactly.
Right?
Yeah. And you know, a lot of the interests that I had as a kid come back as an adult. And so I nurture my creativity through, you know, art and reading and even video games. And I think the fact that I've nurtured my creativity alongside of my scientific prowess really lends to me being a good scientist, right? It allows you to do these creative problem solving.
Absolutely.
And I think the best scientists are well-rounded in that sense.
Yeah, absolutely. And I think one of the frustrating things about science too, because I was a biology undergrad major, but you have to start it when you are a freshman. Like if you want to switch over your junior year, it's so much harder because there's so many prerequisites. Right. So that is another issue.
Like what fraction of kids know what they want to do for their living by the time they're, what, 18, 19?
Yeah, right. They're like, yeah, this is exactly what I want to do.
Yeah. But yeah, and that kind of defines a lot of my even current career. The project ideas that come to you after chatting with a colleague at the coffee shop here or at a conference. I think that if you kind of follow your gut instincts and really just embrace the serendipity of it all, it's really, it leads to more interesting science and more interesting project ideas.
Yeah, absolutely. So can you tell me what you're working on here at Scripps then?
Yeah. So my research in general works on genome editing, and particularly trying to get it useful enough to the point where we can use it inside of the human body or in vivo. And genome editing, what it is, you know, when you have a genetic disease, there is a misspelling in your DNA. And genome editing proteins allow you to go in kind of with this microscopic tweezer or pencil and eraser and physically change your genetic code to correct that misspelling. And what that does is it makes your DNA look like healthy DNA, like nothing ever went wrong. And it also corrects the phenotype, the symptoms of a genetic disease. And so it's one of the few ways where we can actually get curative therapeutics.
So crazy that science has evolved to this point in time. I know. That we're able to do this.
Whenever I tell people, it kind of almost bridges on science fiction.
It does,
which is cool,
absolutely.
Which is another, I think, testing point where you do need to draw a quite rigorous line in between science and fiction, especially with terms of miscommunication. Things like designer babies are not something that I think are going to be realistic in our future. But yeah, so genome editing is something that exploded within the past decade due to the the discovery of something called CRISPR-Cas9. And what CRISPR-Cas9 is it's a protein that's guided by a small RNA molecule to a region of your DNA in your genome. And I like to kind of put this into perspective because I say this kind of nonchalantly, but it's an incredible feat. You have 3 billion base pairs in your genome, and you're able to just write out a sequence on a small RNA molecule, and this protein can target 1 out of those 3 billion base pairs to extreme specificity.
Yeah.
And so it is a very incredible technology to be able to target and manipulate the genome. And so researchers use this protein to target the genome, and they tether on different DNA manipulating enzymes. And so you're able to make all sorts of DNA manipulations, including you can create indels, which leads to gene disruption. So if you have a disease that has overexpression of a gene, you can tune down that expression to fix the gene. You can make small point mutations. So a very large majority of genetic diseases are caused by a single base pair misspelling within your 3 billion base pair genome. And this can have extremely devastating disease phenotypes, but we can go in and very precisely fix that one base pair and change it to the healthy base pair. And so that's kind of, in a nutshell, what genome editing is. But in order, we're starting to really reap the benefits of all of this decade or so of research. So actually quite recently we had a clinical trial that got approval, FDA approval, and this is a drug called Casgevy, where they take patient cells, patients who have sickle cell anemia. They take cells out of their body. They use CRISPR to engineer them to make them, again, similar to a healthy human cells, and they put them back in the body, and this is a curative therapy for sickle cell. And so now this is the first FDA-approved CRISPR-based therapy, and there are many, many more in the pipeline. And it's extremely rewarding being able to hear stories of people whose lives have been directly impacted with stuff that I work on a daily basis. But I guess in terms of what I want to do to help advance this field is there are a few barriers to advancing it to a general therapeutic. And so there's this idea specifically of using CRISPR inside of the human body that's extraordinarily difficult for a variety of reasons. So when you look at the clinical trials that exist now, a majority of them, I think around 70% of them, are ex vivo therapeutics, which means we take, like the sickle cell, we take them out of the body. Yeah, exactly. So you take them out of the body, you edit them and put them back in. That's a lot safer because you can screen the cells, you edit, make sure nothing's wrong before
there's
you put them
no off-target effects, right? Like it's not snipping some other part of your body that you don't want it to.
And so, yeah, you have a lot more to worry about when you're using it inside of the human body. Another really big barrier is delivery. It's probably the biggest barrier. And so you need to be able to get your CRISPR machinery into the human body and in a very efficient way. And one of the biggest barriers is something called first pass metabolism. So if let's say you want to target your heart or your brain, what you're going to do is you're going to inject it into your bloodstream, and everything in your bloodstream first goes straight to your liver through first pass metabolism. And the liver is a very nonspecific tissue where a lot of these vectors where you use to deliver your genome editing machinery gets soaked up by the liver. And so in vivo use we have currently, a majority of them target the liver because there are many diseases that are born in the liver, and we're really good at targeting and editing them. It makes sense that they would be our first go. But when you want to target the heart or the brain or the muscle or anything else, having it recirculate and actually get to that organ is extraordinarily difficult.
Right. And then I can imagine like toxicity is such a big issue there, right? Like
Yes.
if you have to deliver a ton because so much is going to get metabolized
Yes.
in the liver, it's like, oh, can your body actually handle that?
Yeah, you get liver toxicity
Yeah.
then as well. Yeah. And so I think my lab is currently set up to tackle three big areas of what I feel like are barriers towards clinical translation of CRISPR. The first is to make current technologies safer. So CRISPR-Cas9 is extremely useful, but it does have some issues with immunogenicity and stability. And so
Yeah.
if we're able to create highly engineered variants that bypass all of these innate immune responses and these innate degradation responses, we should make something that is less immunogenic and more efficient.
Right, because then your immune system isn't like, this is a foreign substance, we need to attack it and
Exactly.
get rid of it. It's just much more seamless.
Yeah.
Yeah.
Yeah, and so we're engineering components of the CRISPR machinery to enhance this ability. The second is that we're not currently able to make absolutely every genetic change we can make possible. So, like I said, a majority of genetic diseases come from SNPs, but there's also quite a lot that come from very elaborate genomic changes.
Right.
This can be whole gene deletion, whole gene duplication, chromosomal truncation, chromosomal translocation. Like there's quite a wide variety of things that can happen to your genome to create a genetic change. And so the overarching goal of the CRISPR field, specifically on the tool development side, is to be able to make a toolkit of CRISPR genome editors that can theoretically fix all of these, anything.
Right.
And the one thing that we're really struggling with right now is making very large genetic changes.
Yeah.
So inserting whole genes, deleting whole genes. There's a whole subset of, repeat expansion disorders that it would be great to very precisely fix. And so this is another area that my lab is exploring, is how to develop new tools that are able to make these large genetic changes.
Yeah, and like deliver like large molecules essentially,
Exactly.
right? Like massive molecules.
Yes,
Yeah.
yes. Which again, circling back to the delivery, that's
Yeah.
a really hard
Because the
problem.
bigger it is, I'm sure the more that your immune system wants to attack it
Yes.
and views it as a threat.
And just in general, the harder it is to get into a cell, the bigger it is.
Because, it's like so specific what makes it across the cell membrane.
Exactly.
So if you're trying to get in there, it's not easy.
Yeah, and that's a perfect segue because the third area that I'm embarking on in my lab is delivery. So we have, like I said, there's an issue with liver metabolism, but we have some existing vectors that are, at least in mice and somewhat in humans, quite efficient at getting to other tissues. But these vectors have issues with immunogenicity or longevity, leading to off targets. And so my lab is exploring how to enhance the existing vectors that we have through engineering to make them more specific, more safe, and more efficient.
Yeah. And that vector, to figure out what the best vector is, it's like an evolutionary process, right? Where you kind of iterate, determine the best ones through this like evolution, and then amplify those ones that are seemingly going to be the best at getting into the cell. Yeah.
Yeah. So when I say engineering, a lot of times there are multiple forms of engineering one can use to enhance these molecules. You could use rational engineering or computational engineering, or one of my favorite techniques, which I use very heavily in my lab, is directed evolution. And so this is exactly what you're talking about. Is if you have a biomolecule that you hope to enhance towards some property, you can create a selection very akin to Darwinian survival of the fittest, where you create millions of variants. You mutagenize your whatever you want, whether it be a Cas protein or a viral vector, and you subject it to a selection where things that are more fit or more active survive and those that don't die out. And in this case, this is how we can, instead of screening in a 96 well plate, the bacteria cell or the mammalian cell does it for you. They screen millions of variants all at once so that only the best ones survive, and those are the ones that get presented to you. So it's an extremely powerful method of engineering. But on top of that, I do very much appreciate other forms of engineering and also the combination of all of these. So using rational engineering as a starting point for directed evolution, or using computational engineering to complement directed evolution. I think that these are the ways where if you really want to engineer extremely novel function or really powerful function, combining all of these techniques is kind of the way to go.
So your background is in chemistry and chemical biology.
Yeah.
So in terms of the more computational front or the engineering front, how did you gain that skill set? And like you said, this is such a big problem. You can't just use science to fix it, because it's really going to take these multiple disciplines to be able to attack it from all angles.
Yeah, the short story is I didn't ever gain that skill set. This is, again,
you're teaching yourself.
I'm not, actually. This is why I think Scripps is so wonderful. I don't teach myself computation. It's just that there are many people here who are so willing to collaborate. And I would be silly not to use the expertise that's in this institute, because there are people who are like absolutely at the cutting edge of, protein design or molecular modeling, the computational engineering that could be really useful for pipelines into directed evolution. And so I don't actually physically know how to code or how to do this modeling, but the people around me do. And
you came to the right place.
Yes, exactly. And it's an amazing and powerful technique to complex with my expertise, which is directed evolution.
Yeah, that's amazing. So we talked about you as a chemist, or transitioning more to chemistry as an undergrad. What led you more to the gene editing, genome engineering field?
Yeah, also a little bit of spontaneity. So like I said, I started college as a biologist because I do really like biology. I grew to love chemistry, and when I was interested, once I figured out that I really liked research, I had to think long and hard of what I wanted to do with that research long term. And I thought the ideal thing to combine all my interests is, why don't I just smush together the biology interests and the chemistry interests, which means that I was interested in chemical biology. So I applied to a variety of graduate schools that had chemical biology programs. I ended up going to Harvard, and I ended up joining David Liu's lab at Harvard, who is probably one of the major leaders in CRISPR, especially on precision genome editing. And so this was also a little bit of spontaneity, because when I started graduate school, I wanted to challenge myself, right? The only research I did was strict organic chemistry, and the easiest path for me would have been to choose to go directly into an organic chemistry lab for my first rotation. But I challenged myself to do the one thing that was very different from what I did, and I ended up in David Liu's lab. And I don't think he knows this, but when I landed in his lab, I didn't know how to do any of the molecular biology. So I didn't know how to do PCR. I didn't know how to run a gel. I had barely held pipettes in my life. But he took me on, and I think, this learning curve really kind of enhanced my ability to do science.
Absolutely, because you were diving in then, right? You were committing to learning how to do this. You already had this really rich knowledge otherwise too, so I think that also gave you an advantage there.
Yes,
also.
It was hard, and I made a lot of stupid mistakes, but I came in with a very fresh perspective towards the research.
Absolutely, that's so cool. So you mentioned some of the diseases, like sickle cell is an example of that just approved medication. What are some of the other diseases that ideally you would be able to tackle with these technologies that you're developing?
Yeah, so another one which is quite advanced there's a few that are quite advanced in the clinical pipeline. One of them is using CRISPR to make new CAR T therapies. So this is cell therapies specifically to treat cancers. And there's also been success stories, especially young patients with blood cancers.
The blood cancer front, it's amazing. I was just talking to my friends about that over the weekend. Just the great advancements that we're making in hopefully treating metastatic disease eventually.
Yeah, and CAR T therapy has already shown a lot of promise, and where CRISPR comes into this is the ability to further engineer CAR T cells, or more elaborately engineer CAR T cells to make them more potent, or to make them less immunogenic. You can
Right.
even imagine a future where you don't have to, right now what people do is you have to take cells from the patient again to edit them.
And re-engineer them outside and then put them back
But
in.
what if you can have an existing pool of any donor and you can engineer them to be non-immunogenic when you transfer them to a new patient?
Right, so they're just ready to go. Like,
Right.
you don't have to wait for treatment.
So there's a lot of opportunity here with CRISPR. Another really promising area is eye disease, actually. So the eye is a very privileged organ. It's quite easy to engineer, actually, because it's an organ that's right there. You can directly inject into it to deliver your genome editing technology.
Right.
And so a lot of these barriers with delivery don't exist for that eye. And specifically, the eye also lacks a lot of the immune responses for the rest of the body. I mean, it's
interesting.
constantly exposed to
that's
foreign.
true.
So imagine if you constantly had, an immune reaction going in your eye because a little bit of dust got into it.
Right.
But because of that, it's actually quite easy for us to use, and so that's one of the areas where people are pushing forward right now
Right.
is hereditary blindness disorders, and people have actually again been able to see success in this, where patients have their color vision restored after years of lacking the ability to see color.
I can imagine there's so many diseases that there could be hundreds of genes just communicating and we don't necessarily know what the cause is. So is that part of, I'm sure there's so many specific genetic diseases that we do know the specific genetic component that leads to its manifestation, but I imagine there's also so much research that's coming out now that we're now better able to understand how genes might coordinate to eventually cause a disease and lead to even more application in the future.
And that's really tricky because that is true. That gene editing really, as it exists now, requires quite a specific edit to make.
Yeah.
And people can get creative with it. So, for example, in this approved sickle cell drug, you are not actually genome editing the sickle cell allele itself. What you're actually doing is you're disrupting a enhancer that normally turns off fetal hemoglobin. And when you disrupt this fetal hemoglobin, which is a type of hemoglobin that gets downregulated after you pass infancy, it gets upregulated again. And so instead of fixing the gene itself, they're fixing it by supplementing another gene. And so if there are diseases that are too complex for us to fix, like polygenic disorders, etc., there might be a workaround where we can supplement another gene.
Got it. Like a little bit more downstream. Maybe it's like, okay, we know the mechanisms of why this is happening, so we can like tinker with something else a little bit later down the line.
Yeah, I think things like cancer, for example, are
where it's like such a like
complex.
Genetic, environmental, how everything's interacting, right?
Exactly.
It's like, okay, how can we, what's maybe causing this cancer and how can we disrupt that?
Right.
Yeah.
Yeah, and another thing which is really tricky, which is something my lab has thought quite a lot about, is a lot of, there are some diseases where 100% of the patients have this one point mutation.
Right.
And then there are some diseases where it's one gene that's messed up, but within that gene there are hundreds of known mutations that can lead to the same disease. And that's a really tricky problem for CRISPR, because if you think of one gene that has 100 mutations, you need 100 approved guide RNAs to target each and every single one of those individually. And that's just, a FDA nightmare, right? 100 different therapies for one disease. And so one of the huge overarching pie in the sky dreams I have is, how can we make a gene editing technology where we use one treatment to treat all, or at least
fix all of these different components
of these
at
once.
genomes.
And so that streamlines the path to approval, because you only need one individual drug to treat all of these. I think people are a little bit hesitant when you are trying to push forward a therapeutic that might only treat 1/10 of the disease population.
Yeah, when you are trying to tackle these huge problems and you are starting to feel a little bit stuck, how do you get unstuck? Like, what do you do to re-inspire yourself, or maybe problem solve in a different, or see things in a different way?
Right, yeah. I just love taking a break. I'm a huge proponent of mental health. I love my weekend days where I just do nothing, or I take a day trip to go outside. I think that on day to day in science, my brain is my biggest asset, right? And so if my brain isn't up to snuff, then I'm not going to be able to do the research. And so making sure I have those self-care moments are how I relax. And I specifically love and I encourage people to step away from your research when you hit a roadblock, because a lot of times you'll step away and you'll come back after a relaxing weekend and it'll be very clear what either you made a mistake or you overlooked this one area, or it's quite obvious that that direction is the best way to go. And so I'm a huge proponent of relaxation and just stepping away from it all every once in a while.
And I know they even say walking is supposed to just like walking as you're trying to like, you're kind of thinking about it, but you're kind of not. And you're just, giving your brain a break and you're seeing new things and you're not just like focusing on the same thing over again, because you're never going to see anything different at that point.
I'm definitely guilty of the like shower scientific ideas, because same deal. I'm in the shower, I'm relaxing,
distracted,
and then all of a sudden it just comes to me
like,
because
how did I not think about this?
Exactly. It's such a good way. Or when I'm half asleep, of course, obviously my brain just activates and it's like, oh my god, idea. It's frustrating, but that's I think the best way to do it.
That's great advice, especially for everyone in your lab to have that perspective to like take care of yourself, and you're going to do your best work when you're able to do that.
Definitely.
Yeah. I've heard of the people that solve things in their dreams too. I don't know if that's actually, I feel like, I have a lot of engineers in my family, and my uncle was talking about, he's like, I solved the problem in my dream. I'm like, does that actually happen? But
I think for me, it's the opposite. I get scientific anxiety dreams where like in my dream, I ran the wrong PCR and it ruined my whole experiment.
Yeah.
So I'm not there yet, but maybe one day.
That's usually just like offloading the stress, but... So what would you say is the most rewarding aspect of your work, and then also the most challenging?
Yeah, I think the fact that it's so translational is the most rewarding, right? I talked about how now similar science to what I do is in the clinic and even impacting real patients. And so I think having that perspective, it's quite a direct perspective into the significance of what you're doing is very useful for both me and my students. They can go home and tell their family like, hey, this thing that you saw on the news, I work on that.
Right.
And that's rewarding and motivating as well. I've even had people come up to me after talks and, I present data on X disease, and they come up to me and they're like, I don't know if you've ever met someone with X disease, but I'm here and I have to take a pill a day and shots every other week, and it's frustrating, and I'm so excited that my children or my children's children might have a cure for this. And so this is extraordinarily exciting and rewarding.
Yeah, I can imagine. And especially, too, I think you were mentioning how as a biology undergrad, not even really thinking about research or like what you could do outside of medicine, or you're talking about veterinary medicine. So the fact that you can be at the lab bench, but still have, like you were saying, that translational aspect where you can potentially change people's lives, even though technically you're on more of the science side, but you're still making such a difference from that medical perspective, too.
Yeah, and CRISPR is a super unique example of the bench to bedside translation because of how quickly it's made that transition, right? So CRISPR was discovered, like the technology itself was discovered in 2012, and by 2023 we have an FDA approval.
That's insane.
Right? It is, and I actually did a little bit of digging into this. Typically, the timeline between the discovery of a novel technology and when it gets FDA approved, the average is 35 years. And so the fact that we've been able to have this extraordinarily impactful technology that was able to do this feat in 10 years is extraordinary. And it underscores the importance of the technology as well, I think.
Yeah, and like actually developing it to a point where it can do so well in the clinic, and the clinical trials are like more rapid at that point too.
Yeah.
That's so cool. Yeah, I remember, because that's right around when I was starting my biology undergrad. So just kind of like just hearing about it. And now, there's like real applications of it. It's insane.
And in my PhD, David Liu, one of his biggest inventions is something called base editing, which is a type of genome editing which is precision, very precise, and it can, like I said, target a single base pair and change it into another base pair to target these diseases that arise from SNPs. And even in the span of a single PhD, my PhD, the technology was described my first year as a first year incoming student. And by the time I left, it was already in the clinic. It had just entered the clinic. And so it's an extremely unique opportunity as a student, too, to be able to see technology coming from your same lab, which you've been working on for your entire PhD, and you might have even contributed to make that sort of progress.
Right. And I think, too, science and medicine has a bad reputation for being slower, right? Things take time. You want to make sure that they're safe and effective both. We want to figure out how things work. So to not be discouraged by that, to see, oh, no, things can move as quickly as they can, we're living in that time in history, right? Like, things are moving quickly in a way they haven't in the past.
Yeah, hopefully it's getting quicker, so this 35-year time mark starts to decrease, and maybe CRISPR and things like the COVID vaccine are things that break down those barriers, because I think that there are some very important technologies that need to get out to the human race.
Yeah, they need to get to people as soon as possible. So cool. Well, so cool that you're at the forefront of all of this, too, and you're getting to invent these new things and build on what's been done in the past, but just make it even better and change people's lives that way. It's so cool.
Yeah. Doing science changing life.
Yeah, science changing life, exactly. So another thing I want to ask, so Scripps is celebrating its centennial this year. I know you touched on a couple of things that makes this a unique institution, but what's your favorite thing about Scripps?
Yeah, I think Scripps is just such a uniquely curated institute where we're all motivated towards discoveries that change human life. But many different flavors of this, right? We have people that are working on neurological diseases. I do genome editing. There are people that do small molecule therapeutics. There are people that are trying to save the climate. Like, it is extraordinarily diverse. But again, when you get diverse people like this in the same room that are all working towards a reasonably common goal, I think it opens the door for collaboration on a scale that's more innovative than traditionally possible. So you come up with extremely unique answers to very hard problems that you probably wouldn't have figured out if these people have not been brought together.
Right, if you were just in your department of chemistry or you were just in your department of molecular medicine, it's like to have that cross-pollination.
Yes. And I love that, even existing in the Beckman building, the auditorium that's in this building hosts lectures from every single department. And so, it's unlike many other institutes where a lot of times you have your nose to the grindstone in the chemistry department, the auditorium there only hosts chemistry seminars. You're very
siloed kind of.
Yes, yeah. And so the ability to walk downstairs and when a lecture is going on, you don't know whether it's going to be hardcore catalysis or, some preclinical mouse study of treating a neurological disorder or some basic science on XYZ pathway in disease. I really like that, and it really opens up and gives you a refreshing perspective on science.
Yeah, I feel like that's consistently what I hear across the board too. It's definitely very unique in how it encourages that kind of collaboration rather than being more siloed or being more closed off. People actually want to work together across, people want to hear about what other people are working on.
And on top of collaboration, just the inspiration as well. I think that there's a lot of things that people don't even think about until you go to these seminars that are not directly within your field.
And you're like, oh, I'd never, it's like maybe that's the answer to how to solve a big problem that you're working on. Yeah, that's so cool. So if you were going to give advice to an up-and-coming scientist, what would it be?
Yeah, one of the things that has helped me the most throughout my career is not being afraid or embarrassed to ask questions and ask for help. I think that a lot of scientists have this notion of individuality where you have to do everything yourself. You have to do your whole project yourself. But I think it's waste to not use the resources around you. You know, if I am stuck on a particular area of a project, why would I dive deep into a multi-hour literature search of a field that I don't know when someone who is an expert in the field is quite literally next door,
and they could just explain it to you probably better than
exactly. And so I'm never afraid of asking questions or running an idea by a colleague. And I think that greatly enriches how I do my science.
Yeah. Well, and too, I can imagine some people might want to, if they feel like they get help, it's kind of maybe an ownership thing too, right?
They think it diminishes their own.
That's like, oh, this isn't mine because I've had contributors. But it's like, no, we're all working towards such a big common goal.
And when you think of the really impactful science, just how often is it that it's like those one author papers? It's never. It's the one that has so many collaborators listed, but the actual core science is so incredibly impactful.
Absolutely. Yeah, I've never really thought about it that way too. Like, yeah, kind of maybe some of the egos involved in not wanting to ask for help. But it's like, no, we're all going to be better if we can just work together on this.
Yeah, if we could get rid of scientific egos, I think a lot of scientific discovery would go a lot faster.
It's hard, though, when it's like, I'm sure, especially if you've been working on a problem for so long, it's hard to not be, like, so invested in it yourself.
Right.
And so I get that.
Yeah, but I think there is a balance, right? You can ask for help but still maintain ownership.
Yeah, absolutely. That is true. Because exactly. Like you were saying, you can just go next door and ask a question. That's not going to diminish your work.
And especially at Scripps, I think people are so uniquely willing to help. You know, there's people who are world famous who I would never think would give the time of day to a young researcher, and they do.
Yeah.
And they not only give the time of the day, they're incredibly supportive, and they take the time. Yes, exactly.
Yeah, and I think invested in that success.
I think that's wonderful about Scripps and the researchers here.
Amazing. Going back to something we were talking about earlier, how like things you like to do to unwind, like reading and getting outside. What are some of your favorite books?
Oh, I am a very, very big fantasy lover.
Okay.
Right? So the one thing that I think about my hobbies is I really like hobbies that enhance like the creative side of myself. And so that's why when I talked about all of these subjects that I loved as a kid but never ended up going into professionally, they all became my hobbies, right? I paint now.
Oh, so cool.
I'm not good at it, right? But it's so fun to do.
But that makes it better. And I think I took a ceramics class a little while ago, and it was so nice to be able to do something creative with my hands, because like writing is great, but it's such a heady exercise. And so it was so nice and I was terrible, but like to literally like, just do something with your hands and like get out, and like painting is the same thing. It's so visual.
I also recently got into pottery. Like on the wheel, and it's so meditative. And it's wonderful. And then, you know, fantasy books have this element of escapism.
Yeah, I'm the same way.
That I absolutely love. And I also still play video games for the same reason of reading fantasy novels, you know.
Just like getting out of your head. And,
And again, when you change gears so drastically from, you know, every day I'm thinking about science quite analytically, and you go home and you indulge in these curiosity-driven hobbies, I think it resets and rewires your brain in a very positive way.
Yeah, it just enriches it more.
My favorite molecule is the easy answer. It's definitely Cas9. But specifically, the most popular one, which is a Cas9 from Streptococcus pyogenes. And I picked this one for a really unique reason. First of all, I emphasized how amazing it is that it can pick, you know, one single base out of 3 billion.
Right.
And so the activity is just crazy. But the Streptococcus pyogenes Cas9 is the first Cas9 that people discovered and characterized, and it still to this day is the undefeated best.
Is the best. That's crazy that it was the first one.
Right? And there's all this really cool discussion around maybe it was so evidently like out there in the open to be discovered because it's the best, as a bacterial defense mechanism. Maybe it's,
because it was
more fit.
Yeah.
I don't know. But I think that's just really cool. Not often does that happen in a field where your first iteration is just flawless.
Actually the best.
Yeah.
And yeah, 12 years later, like
yes.
you were saying, yeah.
And so Cas9 is definitely my favorite
Best molecule. Okay, awesome.
Best element, I think, would have to be magnesium.
Okay.
Because of how important this is in proteins that have catalytic activity. And so, Cas9 uses magnesium to cleave double-stranded DNA. A lot of proteins that function through ATP use magnesium to be able to be molecular machines.
Right.
So it's just universally a great element in biology.
That's just prolific across everything, yeah.
Yeah. And then in terms of emojis that I would use to describe my research, I spent far too long scrolling through all of the emoji options.
There are a lot of science ones, though.
There are. And so, funny story, in my lab Slack, we actually use the DNA emoji to react to things. So instead of like thumbs up, we're like DNA
I love
emoji.
That. Yeah.
It's so useful in my field. But but I think the easiest answer is a pencil and the DNA emoji, right?
Yeah, you're rewriting. You're rewriting the code.
Yeah.
That's so cool. That actually is very applicable.
Right.
Yeah.
Simple, applicable, obvious.
Yeah, that's so cool.